back to my science lecture channel. Today, we will be discussing the potassium ion channel of Streptomyces A, or also known as the Kcsa. So come on, let's dive in. Hi, hello. Today, we'll be discussing the potassium ion channel of Streptomyces A, or also known as the Kcsa. So we'll be discussing what is Kcsa. Second, what are the parts of Kcsa. Third, what is the process happened in Kcsa. And lastly, what is the function of Kcsa. So now, let us answer question number 1. What is Kcsa? Kcsa, or also known as the potassium ion channel of Streptomyces A, is a prokaryotic potassium channel found in the soil bacteria, also known as the Streptomyces levidans. So the Kcsa is well studied in the said bacteria. So this Kcsa is a pH activated protein channel, which is responsible for the potassium ion gating and shuttling. So the structure of Kcsa was demonstrated using an X-ray crystallography by Roderick Mackinson and its colleagues on 1998. So they explained and demonstrated, or they exhibit the main role of the entry and exit of the potassium ions and some toxin, using the loop and the pore of the Kcsa of the potassium ion protein. The amino acids of this sequence found in the Kcsa is selectively filtered. Also, it is highly conserved. Because the amino acid sequence of this Kcsa can be found or can be dated back in the past or in the old times of science. So as we can see that there is a big importance or relevance in terms of its amino acid sequence, for we can still use it in the study of its evolution and other branches of biology and science. Also, this in this we can study the prokaryotic and eukaryotic potassium ions or potassium ion channel of some of the organisms. So now, we have answered the first question. The first question was, what is Kcsa, or the potassium ion channel of Streptomyces A? Now, let us answer the second question, which is, what is the structural part of potassium ion channel of Streptomyces? So the Kcsa of Streptomyces is an inverted cone in shape. So as you can see from in a cone shape or inverted shape of a cone. So its structure is made up of two helices, the M1 and the M2, and the Kcsa itself is a tetramer, meaning there is a 4 identical single domain subunits, and also it is arranged so that one M2 helix faces the central pore, which is it, while there is also a selective part of the Kcsa. So there is a selectively area of the potassium ion channel. So it is made up of protein also, which it also have a oxygen which helps in selecting or in filtering the entry of the potassium ion into and outside of the cell. Now we have discussed about what is Kcsa, what are the structural part of Kcsa. Now let us answer what is the main function of Kcsa. So the main function is for potassium ion selectivity. So from the word potassium ion channel, so meaning there is a selection of the entry of the potassium ion inside or outside the cell. So through the selective area of the Kcsa, and also due to its helical shape or its helix, it selects the ion or the potassium ion to be specific to inside the cell. Next is the pH-dependent conductase of Kcsa indicates the opening and the closing of the ion channel, which occurs when the protein is exposed to a more acidic environment. So the structure and the parts of the Kcsa it is affected by the pH level of the environment of the cell. Lastly, voltage gating. So, in order to maintain the needed potassium ion of the cell, the structure of the Kcsa transform into a different shape. So, this area will close when there is low level of potassium ion, and it will open if there is a higher level of pH, and also it would open when there is lower amount of potassium inside, and closes when there is higher amount of Kcsa outside. So also there will be an opening if there is a need of potassium ion inside the cell. Lastly, this formation will be changed depending on the environment outside and inside the cell. Also, this Kcsa is located or embedded in the phospholipid bilayer of the cell. For more idea, let us watch this video.
The bacterial potassium channel is a multi-pass transmembrane protein in the plasma membrane. It is built from 4 identical subunits that are arranged symmetrically. A pore in the center of the protein allows selective passage of potassium ions across the membrane. 4 rigid protein loops, one contributed by each subunit, form a selectivity filter at the narrowest part of the pore. This structure is responsible for the channel's high degree of selectivity for potassium ions over sodium ions. In this selectivity filter, carbonyl groups line the walls of the pore. These carbonyl groups are spaced precisely to interact with an unsolvated potassium ion, balancing the energy required to remove its hydration shell. Passage of a sodium ion through the channel is energetically unfavorable because the sodium is too small for optimal interaction with the carbonyl groups.
We have discussed what is Kcsa, what are the parts of Kcsa, what is the function of Kcsa, and now let us discuss what is the importance of Kcsa. So as said that the Kcsa is composed of a highly preserved or conserved amino acid sequence. So the highly preserved or conserved protein sequence is important in studying the more potassium ion channel in other organisms, so that there is an idea, especially during the medical field or in the medical field, and for other field of science, because for example there is a medication that would require potassium treatment. There is an idea on how to attract or how does the potassium enters the cell. So that is our discussion for today, the potassium ion channel of Streptomyces A. I hope you learned something today, and may my learnings and my discussion nourished. Thank you for watching, and for more videos, please subscribe and hit the notification bell. Thank you so much.